Hello and welcome everyone to this video podcast for evolution and diversity. In this video podcast, I'm going to take a slightly different approach. Instead of using the whiteboard, I'm going to use primarily a PowerPoint presentation. As you'll see in this opening picture here, we're going to talk about the diversity of protists. And we've already talked about the many different adaptations that protists have, but now I want to talk to you about the different lineages of protists and some of the features that they have. So let's go ahead and get started. We're going to talk about these 7 different lineages of protists, starting with the Amoebozoa and moving our way down, talking about each of these groups here that I'm circling. And before we talk about each of these, I want to remind you that protists are not a monophyletic group. And so you'll see intermixed with the protists other eukaryotic kingdoms, such as the fungi, the animals, and the land plants. As we talk about each of these different lineages here, I'm going to point out some distinguishing synapomorphies of each of those. Remember, a synapomorphy is something that is present in an ancestral species. So for the Amoebozoa, it would be present in this ancestor, and then present in each of its descendant lineages. So in each of these groups, we'll talk about these distinguishing synapomorphies that unite all of the different lineages within this group. I also want to mention a little bit about the different ways that they reproduce. We won't spend a lot of time on this, but at least I want you to know if it's going to be sexual reproduction or asexual reproduction, if it's haploid or diploid dominant, and so forth. I also want to share with you a few examples, and then in some of these cases I'm going to talk about its relevance. Now this might be ways that we use these particular organisms, some environmental problems that they might cause, and some examples of them serving as a type of pathogen. So let's go ahead and get started with our first one, the Amoebozoa. So again, we have our Amoebozoas here. So we're going to begin by talking about some of these synapomorphies, and then we'll talk about the reproduction and some examples, and then some interesting things about the Amoebozoas. 2 things that distinguish all members of Amoebozoa is that they do not have a cell wall. In addition, they have these lobed pseudopodia that we showed in a previous video when we talked about how they feed and how they move. So these pseudopodia, such as this one here, and all of these that are kind of extending out here, these allow for the amoeba to move in a particular direction and also allows it to gather food and to gain nutrients. I'm not going to say a lot about reproduction with the amoebas, except to tell you that they will undergo both sexual and asexual reproduction. They are single-celled, and as such, they could be haploid dominant or diploid dominant, and we see both types in the amoebas. Okay, now I'd like to talk about some examples of the Amoebozoas. The first lineage here are the amoebas, the loose amoebas, and these are maybe what you're most familiar with when you think about amoebas, these large, massive cells with a very pronounced pseudopodia. Some examples are Entamoeba histolytica and Naegleria fowleri. Another type of Amoebozoa would be the cellular slime modes. And so these exist as single cells that sometimes come together and form a much larger structure, though they are still single-celled. An example of the cellular slime modes are Dictyostelium discoides. And then we have the plasmodial slime modes. These are enormous single cells, much like we see with the loose amoebas here, except that these are even larger. You can see these with the naked eye. And we mentioned these before when we talked about a cell type that has thousands of nuclei in them. So an example of this is the Physarum. Some relevance: there are 2 diseases that are linked to a type of amoeba. The first one is the Entamoeba histolytica that we talked about on the last slide. And this leads to something called amoebic dysentery. Essentially, amoebic dysentery is an infection of the intestines, and they begin to break down the cells of our intestines, and this will lead to severe diarrhea. It's much more common in tropical locations and areas where there's poor sanitation. The way one gets this is by cross-contamination with feces, because this amoeba is found in feces. So if you happen to touch the feces, or if you touch something that had already touched the feces, and then you accidentally touch your mouth, or maybe you drink some water from a contaminated river, or maybe you drink water from a location where there's not very good sanitation. And so this can introduce this amoeba into your system. Now, from what I've read, most often when you consume this, it doesn't lead to amoebic dysentery. About 10 to 20% of the time it will. However, once an individual gets amoebic dysentery, it does have a pretty high mortality rate of 55 to 88%. It's a pretty wide range, and I suspect this range comes from the location where a person is diagnosed with it. But, roughly 40,000 to 100,000 individuals die each year from amoebic dysentery. It can be easily treated if it's caught early. This other one is kind of a terrifying one, I think, anytime you see something that says brain eating. But this is a brain eating amoeba, and it will destroy the neurons of our brains. And again, it's caused by Naegleria fowleri. This amoeba is found in freshwater sources, so rivers, lakes, ponds, rock pits, mud piles. It's not found in the ocean or in salt water, because this amoeba will die in salt water. It's also not found in public swimming pools, if the public swimming pools are properly treated with chemicals such as chlorine. It enters through an individual's nose, and it has a very high mortality rate of 95%. While these types of amoebas are pretty common, they rarely cause sickness. But when they do cause sickness, it is devastating. Something interesting about Dictyostelium is that they usually live as these single-celled organisms. However, when nutrition becomes scarce, these single cells will come together and form these slugs. Now, these aren't slugs like you might think of as being a slug that is a multicellular organism, but rather these are all the cells that come together and they move in a particular direction, like you see in this video here. Then, at some point, what will happen, and you can see it here, they will come together and then they will begin to form a fruiting body that you see down here. And you can see them forming right here, this little one here, and others are extending up like you see here. And as they extend up, at the very top of these, like right here, are the spores, and they will be released and begin to survive again as the single-celled organism. All right, next let's talk about the excavata. And like we did with the Amoebozoa, we're going to talk about some distinguishing synapomorphies. We'll talk about how they reproduce, and then we'll talk about some examples and some relevance. So every member, all of these different lineages of excavata will have some common synapomorphies. Two most noticeable ones are that they have this feeding groove that we see here at the very top of this picture shown here. And essentially what this feeding groove does is it traps bacterial prey. So this protist will eat bacteria. It gets trapped in this groove. It's almost like a big open mouth. And once it gets trapped in here, the bacteria will be taken into the cell, phagocytized by the cell, and in the cell it will be digested, and then the nutrients from that bacterial cell will be taken up and used by these types of protists. Another interesting thing about the excavata is that they lack a typical mitochondria. Now, interestingly, this group once had a mitochondria, but then it lost it. And so it didn't completely lose it. There are still remnants of an organelle that still has some mitochondrial functions, but it doesn't have that massive ATP production function. So this is a really good example of an evolutionary path that took a cell that had a mitochondria, but because it wasn't necessary for their overall fitness, over time, this group of protists lost this mitochondria. Reproduction within this group is primarily asexual. So here are some examples. In the group of parabasalids, there are several examples, but they usually form some kind of symbiotic relationship with their host. As is the case with this example, the Trichomonas vaginalis. This turns out to be a very common sexually transmitted infection. Despite its name, both men and women can have this STI. Now, sometimes they form a symbiotic relationship that is beneficial. In fact, some species of the parabasalids will live in the guts of termites. And in the guts of the termites, they will also have a symbiotic relationship where bacteria live in this protist. And then these bacteria is what allows the termites to consume and eat wood. It's actually the bacteria that live inside the protist that are digesting the wood. In this group, the diplomonads we see this species, the Giardia, which we've talked about before, and they can lead to a gastrointestinal disease called beaver fever. This group also includes the euglenoids, and we've talked about one of their main species, the euglena, before, and that is because they are photosynthetic. And the last group here, the kinetoplastids, they can also lead to various diseases, including Trypanosoma brucei, and this is linked to African sleeping sickness. So as far as relevance goes, we already mentioned some of these. We mentioned the Trypanosomas and how they lead to African sleeping sickness, and the Giardia, which leads to beaver fever, which is a gastrointestinal disease. Though I didn't include it again on this page here, also remember that the parabasalids form the symbiotic relationship within their host, and sometimes it's a beneficial one and sometimes it's a parasitic one. Another relevance, as I mentioned before when I was talking about the euglenoids, is that the euglena are photosynthetic. They have chloroplasts in them. All right, let's next talk about Rhizaria, shown here. And again, I'm going to talk about some synapomorphies that distinguish the different lineages of Rhizaria, talk about its reproduction, and some interesting things about Rhizaria. So first, the synapomorphies that distinguish Rhizaria is that they don't have any cell walls. They also have this interesting calcium carbonate shell, and they have a very unique pseudopodia, as you see in this picture here. They appear thread-like and almost like spikes. Now, the reproduction is usually asexual. That is the most common form of reproduction for Rhizaria. However, there are some that will reproduce sexually, and in the multicellular Rhizaria, we do see some evidence of alternation of generations. Some examples include the foraminiferae, shown here, the actinopods, shown here, chlorarachneophytes, shown here. Now, unlike some of the other protists that we talked about, none of these are known to be linked to any disease. They do, however, form a very extensive sediment on the ocean floor because of that calcium carbonate shell they make. Fossil record shows that Rhizaria species lived at least 530 million years ago. Now, they also play a role in our climate, and you'll probably learn more about this when you take ecology, but they provide an important vector for carbon transport from the surface to the deep sea. In fact, we've seen species of Rhizaria 11,000 meters below sea level. All right, that's all I want to say about Rhizaria. I want to move on to Alveolate right now. And they are down here. And again, we're going to talk about some synapomorphies for them that are be found in all of the lineages. There are some interesting things about this group of protists that I want to highlight, and so let's go ahead and get started. One interesting thing about this group of protists is that they all have sac-like structures that are called alveoli that provide support and located just under the plasma membrane. And so in this picture here, you can see where the alveoli are located. Each of these, they almost look like pillows sitting here. These are alveoli. All of these are. Another uniting synapomorphy for all of this group of protists is that they all move by flagella or cilia. In this picture here, we see cilia shown. Reproduction in the protists is bisexual and asexual reproductive mechanisms. There are three major subgroups for the Alveolata. The dinoflagellates, these are photosynthetic, and they can also be bioluminescent. And we'll talk about those a little bit on the next slide. Another subgroup are the ciliates, and they are covered with cilia, and you can see that here in this picture. An example is the Paramecium caudatum. The third subgroup are the apicomplexans. An example are the Plasmodium, and these all cause disease. They also contain a structure that we talked about in our last class called apicoplasts that are similar to chloroplasts because they arose from a chloroplast, but they no longer have all of the chloroplast functions. Now, dinoflagellates, as we mentioned before, they are photosynthetic, and they also cause red tides, and you can see a picture here of a red tide. This occurs when these dinoflagellates accumulate in large numbers, and they discolor these coastal waters. Now, the danger to these red tides is that they can deplete the oxygen in these areas, and that can lead to various dead zones in these areas where no life can exist. And they can also release toxins that may be linked to various human and other organisms' diseases. And they can also release toxins that are linked to diseases in humans and other organisms. Now, they also do this really cool thing here. At least some of them do. Some of them are capable of having bioluminescence. And here what we see are many of these dinoflagellates that are bioluminescent in this area of the water, and they literally make the water fluoresce because these individual dinoflagellates have an enzyme in them that fluoresces. What's the purpose of it? Well, there are several ideas. One idea is that they serve as a defense mechanism. The light that they produce may prevent them from getting eaten by various predators. Alternatively, they may also produce this light that attract other organisms that will eat the predators of the dinoflagellates. Another thing I'd like to mention are the apicomplexans. As I said earlier, all of the apicomplexans are linked to diseases. One of the most deadly are the Plasmodium, which cause malaria, and they cause malaria in very specific regions, particularly in tropical regions. All right, now let's move on to the Stramenopila, shown here. And like we've done with the others, we're going to talk about the synapomorphies associated with the different groupings of the Stramenopila. The distinguishing synapomorphy found in this group of protists is that they have a pair of flagella that have hollow hair-like projections. So here we see the flagellum here, and then extending from it, you see the hair-like projections coming from the flagellum. Stramenopila, they will reproduce asexually and sexually. When they exist as a haploid, they are dominated by the diploid stage of life. In multicellular Stramenopila, they will exhibit alternation of generations. There are two main groupings that I'd like to talk about. The first are the diatoms, and we've talked about these before because of that glass-like structure they form. And then the brown algae that form these really massive kelp in these large kelp forests. All right, so now let's talk a little bit about the diatoms and the brown algae. They both serve as primary producers because they both contain chloroplasts, and as such they produce oxygen. And they are eaten by primary consumers. That is, they are eaten by various herbivores. Diatoms, as we've talked about before, and you see here, produce these really interesting ornate glass-like shells. Another interesting historical fact about this group is that this particular species, Phytophthora infestans, was the protist that is responsible for the Irish potato famine from 1845 to 1852. Don't worry, I'm not going to ask you a date like this on an exam, but just in case you were curious. But you should know that it caused the Irish potato famine. One other interesting thing about brown algae is that they form these large kelp forests. And because of the size and the vastness of these kelp forests, they end up providing a very diverse habitat for many different animals, as you see in these different pictures. All right, let's move to this next lineage of protists. And let's talk about the group called plantae. And we're going to talk about some of the synapomorphies that unite this group. But one thing I want to remind you of is that this group of plantae includes, as you might suspect, land plants, as the name would imply. But it also includes red algae, green algae, and glaucophyte algae. We're going to spend more time talking about the green plants here, the green algae and the land plants, later on. But we're going to introduce some of the concepts here. Now, one distinguishing synapomorphy for all plantae is that they contain this chloroplast that has a double membrane structure. It arose due to a symbiotic event that we've talked about before, where a cell that contained the mitochondria already engulfed a cyanobacteria that led to all the groups of plantae. They reproduce by both asexual and sexual mechanisms depending upon the species and groups that we're looking at. Some algae, and very importantly, all land plants, exhibit an alternation of generation that we talked about in the last video podcast. And here are some examples: the glaucophyte algae, the red algae, the green algae, and land plants. Okay, so let's say a couple things about the glaucophyte algae. Their chloroplasts, as you see here, appear to be blue. They often live as plankton and are found attached to various substrates in freshwater environments, particularly in bogs and swamps. Now, red algae, as you see here, appear to be red. Now, they're not all red, but most of them are red, and this is primarily because the chloroplasts contain a pigment that appears red. Because of the light that these algae absorb, they can live at a greater depth than some of the green algae. And as I said before, green algae and land plants we're going to cover in a future chapter. Now, all plantae are primary producers, as we've talked about before. They produce oxygen and are eaten by primary consumers. Green algae, as we'll talk about later, they're the closest relatives to land plants. All right, let's move on to our last group that I want to talk about for this podcast, and that is the Opisthokonts. They include the fungi, which we'll talk about later, and the animals, which we'll talk about later. But they do contain this one subgroup of protists. But what unites all of them is this one distinguishing synapomorphy, and that is reproductive cells that contain a flagellum will contain a single flagellum, as you see here. The cristae in the mitochondria are flat, not tubular. And I don't know if we've actually defined what a cristae is, but if this is a cross-section through a mitochondrion here, where this is the outer membrane of the mitochondria, the inner membrane forms these elaborate invaginations of the inner membrane, shown here. And we call these cristae. They are the inner membrane, but they are commonly referred to as cristae. And even though we're talking about them in this group of protists, they also do include the fungi and animals. And remember, that's because the protists do not form this nice single monophyletic group. Now, since we're going to spend a lot of time with this group when we talk about fungi and animals, I'm not going to spend a lot of time right now, except to say that as far as reproduction is concerned, they reproduce by both sexual and asexual mechanisms, depending on the species. The eternoflagellates are the closest living relative to animals. Fungi play an important role as decomposers and as toppings for pizza. And finally, animals play an important role as consumers. Essentially, what I'm saying there is that animals get a lot of credit in the environment and in our ecosystems because we eat things, right? That is, animals will eat primary producers, that is, eat various plants. We'll also eat the animals that eat those plants, right? The primary consumers. And then we'll eat animals that eat those primary consumers, secondary consumers, and so on. Okay, on that note, I think I'm going to end this podcast for today. And if you have any questions at all, please let me know. If not, I will see you in class. Bye for now.